Hey guys, welcome to today's video. Here in this short video, we're going to talk about some inhibitors to ATP synthase. So the four examples I have for you here are DNP, hydrocyanic acid, carbon monoxide, and oligomycin. In the case of DNP, what it does is it basically, so let's imagine actually the intermembrane space of the mitochondria. So this is the outer membrane of the mitochondria, and this is the inner membrane. And we call this space the intermembrane space of the mitochondria. And what DNP does is, well, you'll remember that through active transport of particular proteins, hydrogen ions are transported from the mitochondrial matrix through the inner membrane into the intermembrane space. So we have hydrogen ions here. And DNP makes these hydrogen ions flow back into the mitochondrial matrix without going through the ATP synthase molecule, which is the one that actually makes the ATP, but rather it actually goes through just the phospholipids. And so that's why DNP is not conducive, not great for synthesizing ATP. In the case of hydrocyanic water and carbon monoxide, what it does is it inhibits the electrons from being carried on in the electron transport chain. So you'll remember that again, let's say this is the inner membrane and these are the proteins. And you'll remember that, for example, if this was an electron molecule, they're supposed to flow like this, but hydrocyanic acid and carbon monoxide interferes with this flow, or the transport of this electron in the electron transport chain. Finally, for oligomycin on the right, it interferes directly with the ATP synthase molecule itself. So if DNP again made it so that we couldn't reach, the hydrogen ions didn't reach the ATP synthase and just came back through mitochondrial matrix by slipping through the phospholipid bilayer, for oligomycin, it's interfering with this ATP synthase molecule itself. So these are some types of inhibitors that inhibit ATP synthesis. And I will see you guys in the next video.